Hi everyone, welcome to Hudson River Park's Backyard BioBlitz Diversity Digest. My name is Siddhartha, and thank you for joining me today. So this past week we had the first Hudson River Park Backyard BioBlitz that started on the 8th and continued until yesterday, the 14th. And today's Diversity Digest will be breaking down what was seen in the park during this past week, looking at some of the highlights of what was found by both our staff and by park goers and park users, members of the community. So Hudson River Park is a 4-mile waterfront park that is around 400 acres of estuarine sanctuary along the west side of Manhattan from West 59th Street all the way down to Chambers Street. Now, starting this summer, we launched an iNaturalist project to try and collect different, information about hundreds of species that are in the park. What this means is if you want to get involved, you can for free. If you download the iNaturalist app, either on your phone or you just make an account on the iNaturalist website. Again, totally free. You can upload any picture you take either directly through the app on your smartphone, or you can just take pictures with any camera or phone, upload them to your computer, and then post them to the website using your internet browser. If you missed this BioBlitz or you didn't get a chance to post during it, don't worry, you can always contribute to our growing database. We have an ongoing biodiversity project that will log any observations made in the park using iNaturalist. But what we're going to look at today are what was seen in the past week. So, because this is the first one, really briefly, what is a BioBlitz? A BioBlitz is a week-long snapshot of diversity. Well, we have it for a week long. It can be as short as a day, but we tried to give people a little bit more time, make it a week. And we'll be doing a week-long BioBlitz for this month and then the next 3 months. So the next 3 are going to be August, September, and October. And we'll see how they differ and how they're similar and what species overlap and which ones come and go. You can always check our website, hudsonriverpark.org, for the dates. We have Eventbrite signups to remind you. And we also have iNaturalist tutorials and links to our various project pages on iNaturalist if you want to check out what was seen. I think we're just going to jump right into some of the observations that were made. So what I will say is if you do have any questions, please comment on the live stream. We'll try our best to get to the ones we can, and I'm happy to answer any of them for you. So I am going to share my screen so that you are all able to see the project page. So first of all, I wanted to thank our observers. In addition to park staff, we had 5 observers that I want to thank. DJ Ringer, Bruja, Tabulo, Robert Sorensen, and Daniel Atha for all contributing to this week's observations. We had 45 observations this week, made of 38 different species from the 9 observers, and thank you to our 20 identifiers who helped to make these, most of these, if not the majority, research grade observations and confirmed the identities of our observers. So first of all, congratulations to DJ Ringer for having the most observations in the park. We had 28, and he also identified 23 different species. So some of our favorite pictures that he posted, we have this adorable 2-spotted bumblebee. You can see his abdomen here in this looks almost like a thistle flower. Pardon me, I'm not a plant expert, but we see a really great up-close picture of this bumblebee. Bumblebees and bees in general are incredibly important pollinators. We do see quite a few of them in the park due to all the flowering plants. The detail on this picture is so good. You can see all the little hairs on its body. Fun fact: bees apparently make a noise that, or not a noise, but they do the bee equivalent of going whoop when they bump into each other, which is really cute. Here's another picture from DJ Ringer. This is a dogbane leaf beetle. We loved this one because you could really see the beetle's iridescent carapace from the greens getting into the oranges and reds. And then a final interesting picture from DJ Ringer this week was a tomato plant growing on the pier. This looks like it's just south of Pier 45-46 area in the Greenwich Village, and there is a tomato plant. We don't know how it got here. Could have been any number of situations, so that's kind of interesting to see. Crop plants cropping up, no pun intended, in places that you least expect it, especially on the waterfront of the park. Now, in terms of the species that we saw this week, we had a 5-way tie for the most common, first of which was the monarch butterfly. We do see quite a few monarch butterflies here in the park, and you can really just take a moment to look at that gorgeous picture. This is, again, I believe, DJ Ringer. Now, monarchs we see, especially in the summer and fall. We do have a monarch way station in the park in the Chelsea area, which is our habitat garden, which is all native plants. There is milkweed there, so we obviously see a lot of the juveniles there. But monarchs are one of the more common butterflies we see throughout the park during their migration season. And a great place to look for monarchs, like we said, is the habitat garden, but also anywhere that you have the giant allium plants, like these guys right here. Butterfly bushes, allium, great places to look for monarchs if you're in the park. One of the other really common species we saw this week were blue crabs. So the aquatic animals we had those are mostly from our staff, where we have traps. So you can see these crabs are sitting on a two-tiered crab pot. We have crab pots and minnow traps that park staff check weekly as part of our ongoing fish ecology survey. And what you can see here is actually 2 crabs mating. So you have the male on top. You can see he's a little bigger. He has these huge lateral spikes. That's one of the easy tells that this is a blue crab in addition to its eponymous blue coloring. What's really interesting about this is that blue crabs are incredibly solitary. Blue crabs do not like being around other animals in general, but especially not other blue crabs. In fact, unless it's mating season, any 2 blue crabs, male, female, any combination of sexes, they usually try to kill each other. They're incredibly aggressive predators that eat basically anything that they can get their claws on. It's thought about 13% of their diet is actually other blue crabs. Not terribly uncommon in aquatic organisms and crustaceans, but fewer are as aggressive as the blue crabs are. So mating is actually one of the only times that blue crabs come together in a relatively non-violent way. Because we have so many blue crabs actually breeding in our estuarine sanctuary, they are definitely one of the most numerous organisms caught in our traps. We caught 750 juveniles last year alone, and that's not counting the couple hundred adults that we also caught. All right, moving on to another, we had monarchs, blue crabs, oyster toadfish. So oyster toadfish, I love this picture. This was from our staff, Olivia, one of our educators. This is definitely our most caught fish in our fish survey. Part of that is because we are using traps. We're not using rod and reel. These are benthic fish. They live on the bottom of the river. Their whole body, you can see, is adapted for that. They have eyes situated on the top of their head, sort of like a crocodile or an alligator, because they are submerged ambush predators. Not that they're peeking their heads out of the water, but they're low on the bottom looking for prey to swim by, and then they'll surprise them and get them with those huge, huge jaws. This is definitely an adult. You can see it has a bit of scarring on its right jaw here. They're incredibly tough fish. They can, even though they don't have scales, they're really hardy. They have a lot of muscle. They're mostly jaws with a tail. You can even see the spines here off the side of the operculum that also help to protect them. We caught around 350, 400 toadfish. No, 200, sorry. 150 to 200 toadfish last year, which was out of our 400 fish caught last year. So they're a pretty substantial majority of the fish that we do catch in the park. They're here year-round. They're an estuarine species. You don't see too many as soon as you start getting too far up or too far back out to the ocean. And they're called oyster toadfish not just because they look like toads, but they also sound like them. They croak. They use their swim bladder to produce a croaking sound. The males in particular do this to attract the females, but the females also can call in response, and both males and females will call or croak when they're agitated, both in and out of the water. So they're some of our staff favorite fish. So not surprising that we're seeing a bunch of them, especially now that it's getting warmer. This is another really neat fish. So actually, two of our most seen species in the park this week were both syngnathids. Syngnathids are the family that encompass pipefish and seahorses. The family Syngnathidae means together jaw. So they have fused jaws instead of like a normal fish, which opens its jaws like that. This one can only open at the end because the jaws are fused up until that point. So this is a really beautiful picture of our northern pipefish in our educator Toland Kister's hands. So you can see how its head is actually very similar to that of a seahorse. They're in the same family. They're pretty closely related. That makes sense. Here, all similarities depart. It is a longate serpentine as opposed to the classic S-shaped, rigid, upright seahorse body. But they are very similar in a lot of ways. And one of those ways is that the males have brood pouches. So this right here is our male, one of our two seahorses, one of the couple seahorses we caught this week. This is a gravid. We call it gravid as opposed to pregnant because they do not have a womb. Gravid male seahorse. You can see that his brood pouch is incredibly full of eggs right here. And again, you can see that snout with the fused jaws at the front, similar to the pipefish, but then it has this rigid S-shaped body. Now, seahorses and pipefish, again, both do this. The males receive the eggs from the female. She lays her eggs into his pouch with an ovipositor, an egg tube, basically. And then he holds onto them and fertilizes them in his pouch and waits for the eggs to be ready to hatch, and then he'll expulse them from his brood pouch. It's pretty spectacular to watch. And you have hundreds and hundreds, if not more, of baby seahorses that look like basically adults, just very, very small, which is unique because most fish, when they start off as larvae, look totally different. They look like tiny little heads with a yolk sac swimming around. The seahorses come out a little more formed. Seahorses and pipefish are fascinating because, again, like toadfish, they don't have scales. Instead, they have bone plates or bone rings. Seahorses, you can sort of see these, they look like little spikes. Those aren't just spikes, though. They're actually, and rings is maybe the wrong word because they're actually quadrilateral or rectangular rings, for lack of a better word, that make up a serial chain of a secondary skeleton called a dermal skeleton that basically functions like an armor, right? It's a second skeleton around its normal fish skeleton. And this is, the seahorse's tail is actually one of the only scenarios in nature where a square appears organically. Squares are not normally organic structures, but seahorse tails are actually one of the few examples of that, which is very, very cool. So always great to see the seahorses coming in the late spring, early summer, and even better to see them reproducing, which is why they're here and why so many fish come here to the park, is to have babies in the relative safety of the shallow, murky water. It helps protect them from larger predators. So those are our most common species, but I wanted to highlight another few pictures that some other participants took. This is a great picture of a common tern, I believe taken by Robert Sorensen. Beautiful picture. You can really see the profile of the bird. Terns are one of the easier waterfowl to identify. We have about 17 different species of seagulls, and that can get tricky unless you really know what you're doing. And you can see what this tern is doing is it's resting on the side of a pier. So a lot of water birds use the park not just to hunt, but also to nest, lay eggs. You can see the piles out in the water further in the background there. A lot of cormorants, terns, gulls will rest there once they've found some fish, didn't find some fish, need to take a breather, sit down, recuperate, and then go back out hunting again. We've had geese make nests on these piers. This is actually the air vent to the Holland Tunnel pier, the Pier 34, I believe, on the south side. And you can see there also that this is the same tern. This picture is basically being taken from the ledge where the tern just was, so it hops down onto the lower fendering, possibly scanning the water for fish, possibly just to change venues. So So that's a really nice picture that we had of that tern. And then finally, I wanted to take a look at some of the other non-animal pictures we had. We had quite a few mushroom pictures from our staff, but also one from one of the participants. And these are some type of field mushroom, some agaric field mushrooms. I'm by no means a mushroom expert. We are still working to identify the exact species. So if you do know mushrooms and you have a suggestion, that's the great thing about iNaturalist is you can log on and you can suggest identities for any observations around the world. So definitely encourage anyone who's interested to, even if you don't want to go out, even if you don't want to take pictures, you can always help out by identifying them. These are really, really neat. So we actually have mushrooms pop up in a lot of places in the park, sometimes even in places you wouldn't expect, like gravel, like picnic yard areas. You'll see them popping up in some of the cracks, sort of like the tomato plant popped up on the edge of the pier. But yeah, these are really neat mushrooms. And actually there's another, yeah, here's another shot. You can see here's the top of the cap. We have the gills underneath. They're called the gills or lamellae. Here's the skirt or the ring of the mushroom, and then the stalk, obviously, supporting it. The gills usually are where the spores come from. The spores are emitted from the underside and then are transmitted in as various ways as there are species of mushroom, which are many. So those are most of our pictures that I wanted to share. We definitely had more that are really cool, so I do encourage everyone to check out the July 2020 BioBlitz project page to see all of our pictures of all of the different animals that we saw. There are other insect species, other plant species, other fish species we saw. Again, total of 38 different species. We only went through about 10 of them. So definitely check that out. And I think now we'd like to hear from you before we start taking up too long. So we have a couple questions here. So the first one is, how can I get involved? We talked about that a little bit. Again, if you go to hudsonriverpark.org and you go to the Estuary Lab, our website will be changing very shortly, probably next week. We will be the Hudson River Park River Project, but all the links will be just only more legible. And there we have tutorials, we have step-by-step instructions for how to get involved, how to make an account, how to use iNaturalist. We have a tutorial video. Here's a question: will the tomato plant sustain the salinity? Will be able to survive with the salinity from the estuary? And actually, you might not expect this, but yes, tomato plants are, interestingly enough, surprisingly hardy when it comes to salt. They can typically tolerate coastal area salinities. This is something we see, and this goes into a lot of urban planning, is, when you're planting things near the water, you do have to account for the salinity of the soil. The salinity that will condense from the air as well can sometimes not work with some plants. I'm not sure if it's the same word for plants, but in terms of animals, the word we would use is euryhaline, which literally means wide salt. It has a wide salt tolerance, as opposed to stenohaline, which is narrow salt tolerance. And then our last question we have is, where are good places to see birds? Oh, and this also reminds me, another great place to look at monarchs is Piers 45 and 46. So Habitat Garden, Piers 45 and 46. And then anywhere that you see butterfly bushes or the giant allium, those big purple plants. Our last question is, right, where can I find birds? Where's good places for bird watching? I would recommend the Pier 32 boardwalk, so actually right near, just south of where that tern photo was taken. And actually the pile field you saw in the background of that photo is a great place. We see gulls, we see cormorants drying their wings there all the time, a lot of waterfowl, a lot of ducks, mallards and such in the water. I think I've seen a merganser or two as well. And then additionally, Chelsea Waterside Park, the Chelsea Bowl, is a great area to see a lot of perching birds because they're all in the trees in that area. So if you have any other questions, please, you can feel free to reach out to education@hrpt.ny.gov. With them, we'll be happy to answer them. We hope you enjoyed this first BioBlitz and Diversity Digest. Our next one will start August 12th and will run until Tuesday the 18th, so a week from Wednesday the 12th to the following Tuesday the 18th, with the digest on the following Wednesday the 19th. So just like we had this week, it'll be exactly the same. So if you're interested in other programming that we're doing live tomorrow at the same time at 2 p.m., we have our weekly live from the field. We will be looking at enterococcus pathogen testing in water. And so you can join myself and some of my colleagues looking at how we test the waters in the park for pathogens that are indicative of sewage pollution. So thank you for joining us. And oh, there's one other thing. We have a couple other events coming up. We have not just live from the field this week, but we also have next week on Wednesday, 2 p.m. We have our first in our lecture series of Ask a Scientist. We will be joined by Dr. Peter Park, who is an evolutionary ichthyologist, community angler. He runs fishing programs, and he also teaches about education theory at Nyack College. He's a wonderful, wonderful fish biologist, a great teacher, and he'll be speaking about community angling, fish in general in the Hudson River and East Rivers, where he does work out of. And you'll also be able to engage with and ask him any questions you might have about any of the subjects that he's talking about, or just fish ecology and fish biology in general. So thank you, everyone. If you've joined us, then please keep joining us if you enjoyed it, and looking forward to see what creatures you find in the park.